about possible usage of DNA-abzymes as biomarkers of systemic lupus erythematosus and rheumatoid arthritis, or at least I'm going to try. Systemic lupus erythematosus and rheumatoid arthritis have very, very different symptoms. There are lesions of skin, of pleura, of pericardium, of joints, and sure, there are vasculitis and glomerulonephritis, and all the others. And pretty often when there is the bright clinical manifestation of the disease, it's pretty often too late for the treatment with the full recovery. And that is the problem. But fortunately, in 1957, a group of scientists with the head of Cipollini realized that there is some kind of reactivity of sera of patients with lupus and DNA. And 30 years later, another group of scientists realized that this reactivity was due to DNA-abzymes. So DNA-abzymes are the molecules which have two main roles. It can be like antibody with cytotoxic antibody with catalytic activity. And in theory, there are two functions. It hydrolyzes DNA and it have cytotoxic reactivity for the cells of tumor lines. Oops. But how come that these DNA-abzymes appear in the sera of patients with lupus and rheumatoid arthritis? So it can be due to dysfunction of immune system. Well, sure, this reason is quite general, but anyway, it can be due to dysregulation of different genes. For example, the genes which are responsible for production of interferon 5, which can turn on and turn off macrophages. Also due to dysregulation of genes which are responsible for B cell receptors and for the maturation of T cells and all the others. Another weighty reason of this autoimmunity is that in the bloodstream there are too many apoptotic nucleosomes. And sure, this can happen due to dysregulation of the genes which are responsible for the removal from the bloodstreams of these apoptotic nucleotides. Such genes are, for example, tyrosine kinase mer and serum amyloid protein P and others. And also, sure, B cells can also contribute to the disease, and also in antibody-independent way. For example, because they can produce some pro-inflammatory factors, T cells can do the same thing. So thus, interacting with T cells and B cells, they enhance this autoimmunity, and as the result, there is manifestation of the disease. So, okay, so this way or another way, but these DNA-abzymes, they appear in the serum of people with autoimmune disorders. But why do they appear there? Possibly because it could be like a part of a body's viral and bacterial protective mechanisms of the organism in order to remove foreign DNA from the organism. But obviously antibodies in autoimmune disorders can play three important roles. They can be protective, they can be pathogenic, and they can be predictive. So DNA-abzymes can play both roles, two roles. They can be pathogenic, and sure, they can be predictive. Why they can be pathogenic? Well, first of all, because they have this catalytic activity, and they can hydrolyze DNA. They can connect with some surface markers, for example, myosin 1, then they incorporate into the cell, then they translocate to the nucleus, where they bind to DNA and hydrolyzes it. And they can also connect with the death cell receptors on the surface of the cell. And sure, they can indirectly play a role in pathogenesis because of deposition of immune circulating complexes. So, what can be, why they can be predictive biomarkers? Apparently, there are some kind of researchers which say that almost 80% of people who are with diagnosis of systemic lupus erythematosus have these DNA-abzymes in their serum. And DNA-abzymes, they can be two types, like double-stranded DNA-abzyme and single-stranded DNA-abzyme. And some thought that the double-stranded DNA-abzyme is more specific and single-stranded DNA-abzyme is less specific. But, well, the problem is even that double-stranded DNA, some of clones can be pathogenic and some can be not pathogenic. So, and also what is a benefit of these DNA-abzymes, that recently it was reported that single-stranded DNA have a very high level in the patients with a flare of symptoms. So like it can be a kind of predictive biomarker. And later there was another report which said the same thing. But anyway, there are still too many questions not clear and open, and especially as for the model of like ideal biomarker. And actually DNA-abzymes, yeah, sure, you can find them like in accessible fluids, because in serum, but the problem is also the method of detection of these biomarkers. For example, methods which are quite widespread right now, like Alisa, for example, or modification of Alisa with deoxygenin. It's good for screening, but unfortunately it has too low sensitivity. There is another method for detection of these molecules, but for example, FAR assay, but it's radioactive, and you can use it only in the patients who already have the diagnosis of systemic lupus erythematosus, just in order to monitor the disease. Well, but, oops, sorry. So, and sure, it would be quite good to find the disease like on the very first steps, like when it's benign autoimmunity or maybe pathogenic autoimmunity, but before clinical manifestation. So anyway, these antibodies can be used in the future with research sure as biomarkers of autoimmune disorders. That's all.
Thank you so much, Alisa. Thank you. Any questions addressing the speaker?
Well, sure, not I was who measured it. It is just kind of review. And the people who did this report, they used different kinds of detection. For example, it also depends because they needed to pure and to separate like anti-stranded and, single-stranded anti-DNA and double-stranded. So they used sometimes these FAR assay or Alisa, or the one also like Alisa, but with usage of some bacterial DNA. And so also some magnetic fluorescent methods. Yeah, sure, unfortunately, because right now there is no opportunity. Because I'm a student, so
You can have a separate talk on the subject. So much interesting. Including the engineered antibodies. You would have it. We would send you the publications. Yeah.
Yeah, I can give you the link right in a second. Yeah,
Yeah.
sure. Thank you.
Any more questions? Thank you so much.